Hi everyone, I'm Emma Thompson. I'm here with Joel MacKay, who is a professor in biochemistry within the School of Life and Environmental Science. Hi Joel, how are you doing?
Good.
Thank you very much for letting us interview you today. So can you tell me what first got you interested in biology?
Well, I guess my undergraduate degree was chemistry rather than biology, so I did almost no biology other than first year biology as an undergraduate. My PhD was in the chemistry department, so doing organic chemistry, physical organic chemistry. And so I think my interest in biology really didn't arise until later in my PhD, where it started to become clear to me that understanding biology really requires an understanding of chemistry. And so applying the knowledge that I had in the area of chemistry to understanding biology seemed like a nice challenge to me.
A logical thing to do.
Yeah, that's right.
So you kind of made that switch. Was it difficult to make that switch, or did it just kind of evolve?
No, I mean, because my PhD was focused on understanding the mechanisms by which antibiotics act, we were already, looking at biology, but really very much from a chemical perspective. So, the work was important now for understanding how different classes of antibiotics act on the bacterial cell wall. But really, we were looking from a real molecular perspective, and I think it was that kind of made me appreciate that, as I say, that understanding biology relies on an understanding of chemistry. Yeah.
Wow. So already sort of multidisciplinary there. So do you still do work on antibiotics now?
No, I haven't really worked in that area since my PhD.
Okay.
Yeah.
So what is the research that you're doing at the moment?
So for pretty much my whole independent research career, the main focus of my work has been understanding the mechanisms that underlie eukaryotic gene regulation. So how are genes turned on and off?
Yeah.
How is it done at different times in different cells in response to different stimuli?
And do you work in one particular area of genes, or is it just or one species, for instance, or is it across everything and as many genes as you can get your hands on?
So the thing that we're interested in are the mechanisms. So the mechanisms that control how genes are turned on and off. They're the same mechanisms irrespective of what sort of cell type you're talking about and what sort of organism you're talking about, at least if you're talking about complex organisms, multicellular organisms.
Yeah.
So the lessons that you learn from yeast and C. elegans are just as applicable to understanding human and mammalian gene regulation as they are to understanding the worms and the yeast.
Fantastic. So what are you specifically working on at the moment?
So one of the main focuses of the lab at the moment is understanding the mechanisms by which chromatin is opened and closed. So you know that DNA is wrapped up in histones, and those histones are wrapped up into larger sort of multi-layered structures, and that's the way that DNA is packaged into the cell. And the opening and closing of that chromatin structure is what's required if a gene is to be able to be turned on and transcribed, or if you need to repair a gene, a portion of the DNA that's been damaged by mutation or by some other chemical insult, or if you want to if you want to transcribe the DNA, if you want to repair the DNA, or if you want to do any other aspect of kind of biology on the genome, then you need to be able to move these nucleosomes around. You need to be able to move around the DNA and the way it's packaged by these nucleosome octamers of histones. And so there's a whole class of enzymes whose function it is to manipulate the chromatin in this way, to shift nucleosomes around, to swap histones for other histones, to put on and take off covalent modifications on both the DNA and the histone proteins that act as signals to turn the expression of the associated genes on or off. So we're interested in one particular protein complex, a multi-protein complex that acts on the DNA both to remodel the nucleosomes and to add and to remove covalent modifications from the histone proteins that package the DNA. And we want to understand how this complex does this right down at the molecular level, because it's only by understanding mechanistically how this complex works that will be able to understand diseases like cancer, which in many cases are caused by problems with the activity of this particular complex.
Wow. So you talk about needing to know these things at molecular level before you then move on to sort of a whole genome of a species. So you've already gone from chemistry into biochemistry, so including that biology in there, and then there's sort of, I guess, an evolutionary aspect to this, and also you're looking at more genome basis, genome wide things. So do you work collaboratively with lots of different people across an entire range of biological disciplines?
Yeah, so mostly the collaborations that we have are with molecular and cell biologists. Okay. So our expertise is very much at the protein structure and biochemical function end of things. And then even as you move into doing cell culture, mammalian cell culture for example, and manipulating cells rather than manipulating purified proteins, which is what we tend to be focused on, then it's a different set of skills. Now we do some of that kind of work in our lab, but more often we collaborate with people that are cell biologists and molecular biologists.
So the experts in that area collaborate with you guys, who are the experts in this area.
Yeah. And at the other end we collaborate to some degree with synthetic organic chemists who can make small organic molecules that in one project, at least at the moment, we're interested in trying to make small molecules that inhibit the function of one of these chromatin regulatory proteins. And so we work with the chemists to make the molecules, and then we're working on how the proteins bind to these molecules and what their structure and biochemical function is, and then we work with the cell biologists to, in that case, for example, to test how these small molecules would actually act on a cell. What would the phenotype be if you treat the cells with these molecules and so on.
And so working within this research environment here at Sydney University, do you work sort of with other research institutes such as the Garvan, and do you also have an applicability with industry that's outside the research area? I
mean, our strongest collaborations are with medical research institutes and universities outside of Australia, actually.
Okay.
So our strongest collaboration is with a chap called Gerd Blobel at the Children's Hospital of Philadelphia, so he's a molecular cell biologist interested in mechanisms of gene regulation, and he works at that kind of next level up of complexity of cell biology and animal testing as well. So he wants to understand these mechanisms at that level, and his interactions with us allow him to, to make hypotheses based on our molecular studies that he can then test in cells, in animals, and so forth. Yeah, so that's one of our main collaborations. We have other collaborations that are like that as well. Yeah.
So predominantly sort of the medical side of things.
That's right, almost exclusively. Yeah.
And then what about applicability to the general public? Does it have that? Once you've done this research and you find these things out, how does that therefore then impact on the global population?
Right. So the main focus of the work, as I say, is understanding the mechanisms by which genes are regulated. Many diseases are caused by problems with gene regulation. So cancer at its very heart is probably the best example that almost all cancers are caused by dysregulation of certain genes, overexpression or underexpression of particular genes. And in many cases the problems are mutations in the kinds of proteins that control gene expression, for example transcription factors. And so understanding how these transcription factors work is really the first step to understanding what you might do to treat problems that arise from these transcription factors doing things that they're not supposed to be doing. So, even though we aren't, in most cases, most of the work we do is not explicitly designing therapies for diseases, although we do some work like that. Most of the work is to form a foundation from which other more translational research might be built. So most of the work is not directly translational, but as I say, as a foundation from which other people would do more translational work.
And in something like cancer, it has a huge, massive implication for a worldwide audience, for want of a better word. So.
And a collaboration that we're just starting at the moment with a chap from CSIRO is focused on transcription factors that are involved in the transition that insects make from larvae to adult insects. So hormones like juvenile hormone are important for regulating that transition. And again, these juvenile hormones act by regulating the transcription of a whole set of target genes. And so again, by understanding how juvenile hormone and molecules like that bind to their target proteins, then you've got a better understanding of the processes that are involved in this metamorphosis. And also you get the potential to design molecules that can either inhibit or enhance the activity of molecules like juvenile hormone if you want to, for example, suppress the metamorphosis of pest insects in agriculture from going from larvae to the adult, or vice versa. You might want to enhance that process.
That sounds like great work.
Thanks.